Elements of transferable antibiotic drug resistance. Part 1. Transfer of genes mediating resistance to commonly used drugs can be intrinsic, that is natural, or acquired through genetic means. It includes chromosomes and transferable genetic elements. Chromosomes may undergo mutation and spread resistance via cell division, also known as vertical mode of transmission. Transferable genetic elements include plasmids, bacteriophage, transposons, and integrons. They directly transfer the resistance via conjugation, transduction, and transformation, that is, horizontal mode of transmission. In this video of part 1, I will mainly focus on plasmids and bacteriophage. For other transferable drug resistance elements, do refer part 2 of the same. Detailed explanation about mutation has been discussed in my earlier educational video titled as Bacterial Genetics and the Mechanism of Genetic Variation. You may go through it. Elements of transferable drug resistance includes plasmids, bacteriophages, transposable elements like transposons, insertional sequences, and integrons. These elements play a critical role in the spread of antibiotic resistance among bacteria by acting as platforms where antibiotic resistance genes are located. Term plasmid was first introduced by Joshua Lederberg in 1952. Plasmids are extrachromosomal, mobile, autonomously replicating, covalently closed double-stranded DNA, mostly circular. Size may range from 1 to more than 400 kbp. Serve as a scaffold on which arrays of antibiotic resistance genes are assembled. They also serve as an agent of genetic exchange, mainly by conjugation. Plasmids involved in resistance genes dissemination are known as R factor. R factors is composed of two functionally distinct parts. Resistance transfer factor, that is RTF. It includes origin of replication and genes facilitating their sustainability in host bacteria. Genes transfer multiple drug resistance among bacteria. And resistance determinant, that is R region, carries several drug resistance genes. Types of plasmids based on their transferable property includes conjugative plasmids and non-conjugative plasmid. Conjugative plasmid is a primary factor in the emergence and increase in multiple drug resistance in bacteria. Non-conjugative plasmids can further be categorized into mobilizable or non-mobilizable. Mobilizable can get transferred if the conjugative function are provided by a separate self-transmissible F plasmid carried within the same bacteria. Non-mobilizable cannot induce conjugative mating and are not transferred between the bacteria. F plasmids contain the genes needed for the process of conjugation in bacteria. Bacteria can become resistant to various antibiotics either by the acquisition of several independent plasmids or through acquiring a single plasmid with many resistance genes on it. Plasmid mediated antibiotic resistance. Beta-lactamases break down the beta-lactam ring to an inactive form in case of beta-lactams. Aminoglycosides modifying enzymes like acetyl transferases, phosphotransferases, and nucleotide transferases in case of aminoglycosides. Alteration of cell membrane permeability to the antibiotic in case of aminoglycosides hinders binding of the drug or causing their dissociation from ribosome in case of tetracyclines. Induced enzymatic activity due to methylation of 23S ribosomal RNA in case of macrolates. actively expose the drug out of the bacteria in case of sulfonamides, acetylation of the antibiotic to an inactive form in case of chloramphenicol. Bacteriophages. These are also known as viruses of bacteria. Detailed features of bacteriophage, their types, their classification, their mode of replication, and their significance has been discussed in my early uploaded video titled Bacteriophage. Phage infecting the bacteria can either follow lysogenic cycle or lytic cycle. In short, lysogenic cycle incorporates phage genome into the host genome and replicating as a part of the host. Lytic cycle, the phage multiply inside the host cell and release new phage particles after cell lysis. Transduction of bacteriophages includes any sort of bacterial DNA, including linear chromosome fragments and all sorts of mobile elements such as plasmids, transposons, and insertion elements. Dissemination of drug resistance genes occur via generalized transduction and specialized transduction. In case of generalized transduction, transfer of any portion of the donor genome to the recipient cell takes place, either by lytic or lysogenic phage. In case of specialized transduction, it involves only template phage, produces particles carrying both chromosomal and phage DNA, and transfer only specific regions of the bacterial chromosome. Phage mobilize antibiotic resistance genes through generalized transduction. Detailed explanation about conjugation and transduction has been discussed in my earlier uploaded video titled Bacterial Genetics and Mechanism of Genetic Variation. In nature, concentration of bacteria and bacteriophages are high enough to guarantee a great frequency of their encounters that are needed for transduction to occur. Due to structural characteristics, phages are more stable in their extracellular phase than that of free DNA, which is more sensitive to nucleases, temperature, and radiation. This higher survival of phage DNA makes them suitable candidate for transferring genes among bacteria in the environment. Bacteriophage mediated transfer might be crucial in the mobilization and transfer of chromosomally located antibiotic resistance genes of environmental bacteria to human and animal pathogens. In vitro, phages transduce resistance to imipenem, aztreonam, and ceftazidime in Pseudomonas aeruginosa by generalized transduction. Strain Salmonella enterica 0 typhimurium DT104, characterized by various multiple resistance patterns, transduces some of the resistance genes. Bacillus anthracis template phage encodes fosfomycin resistance. Since 1970s, evidence presented that prophages participate in the dissemination of erythromycin resistant streptococcus infections. Study of phage DNA from different hospital and urban treated effluents showed the presence of high level of genes conferring resistance to beta-lactam antibiotics as well as genes conferring reduced susceptibility to fluoroquinolones. Thank you for watching. Do subscribe to the channel and please hit the notification bell to remain updated with part 2 of the same video.